can continue our exciting work today with the participation of Professor Shamil Sunyaev. TÜBİTAK Research Institute for Fundamental Sciences organizes Scientific Türkiye public lecture series with the participation of the distinguished scientists and great speakers of the world. The aim of this event is to share the passion for science and research with a wide audience, as well as to foster scientific curiosity and scientific culture in the society. As Einstein said, the important thing is not to stop question. Curiosity has its own reason for existence. Professor Shamil Sunyaev from Harvard University has kindly agreed to give a public lecture as part of this lecture series of TÜBİTAK researchers. He is going to give a great talk about causes and consequences of our genetic diversity. Please have the pleasure of listening to Professor Shamil Sunyaev, and I have a pleasure of inviting him to this stage.
Okay, well, thank you for, I think most of you continue with today. I wasn't sure about the attendance for the public lecture and the format. So for those of you who attend the scientific lectures, a lot of this is going to be redundant. And presented in a more accessible fashion. I hope that's okay. And I will talk about primarily the same things as we discussed this morning, and we're going to discuss tomorrow. But okay, the whole point of this discussion is what causes and what happens with human genetic diversity. So we all know that humans are completely identical. Some people are twins, but still not completely identical. And a lot of these differences are genetic. And again, this is the Galton plot, very classic early 20th century study where height of healthy child is plotted against mean parental height. And we know that the children of taller parents are on average taller, and children of shorter parents are on average shorter. So we think that a lot of these height differences are heritable. And in fact, a careful look at it shows that about 80% of differences in height are genetic. Right? So a lot of this diversity is genetic. What happens at the molecular level? So now we have technology to sequence DNA. We can peek at individual nucleotides. And what we see is that if we think about DNA as a sequence of letters, so here's G, C, and T, and every element here represents one letter of the alphabet, or one DNA base, some of them are different between two randomly chosen chromosomes. And not even between individuals, right? So I have two complementary sets of chromosomes. One is inherited from my mom and one is inherited from my dad. And I have about one letter different for roughly 1,000 bases. That's the source of diversity even within one individual. It's not only differences in individual letters. We have what we call insertions and deletions, where a few subset of the sequence can be inserted into the sequence or deleted from the sequence. So we have some letters are missing, and some letters are being added to DNA. There are large events that are called structural variations, where larger chunks of DNA may be missing, or may be duplicated, or may be inverted. all of this more complicated events. I will primarily talk about singular variation, but you should keep in mind that all of this collectively make human differ from each other. Okay, why is this interesting? Maintenance and transmission, fair transmission of genetic information across generation is an amazing fundamental property of life. It's maintained through two processes. So one is fidelity of DNA replication. At every cell division, DNA is being copied. So there are two identical new copies of the molecules created from one. And this process is extremely accurate. Right? It's very high precision copying process from one molecule to the other. On the other side, DNA is subject to a lot of damage, right? So there are various chemical agents, radiation, whatever is happening to DNA that makes various chemical changes in the sequence. And DNA is being constantly repaired. We have multiple repair systems that fix DNA back to what it should be. Again, this is a very efficient system. Very efficient system. Still, there are mistakes. And mistakes happen at the time of copying DNA, like at the time of replication. When one DNA molecule gives rise to two other molecules, sometimes there is a wrong letter being inserted. Also, some DNA damage is either not repaired in time when you have to divide, and then most systems introduce mutation at this point, or this repair process is incorrect. So it told me it's highly efficient, but occasional error still is there. So this leads to changes in the DNA sequence and propagate into differences between individuals. How many differences? So we have roughly about 70 changes if I talk about point mutations, so changes of individual letter. Some people have 100, some people have as few as 40 per individual individual. Right, so again, these are not variants inherited from mom and dad. These are completely new events in the sequence. So there is continuous influx of this into the human population. Right? So everybody carries some of this event. This is our own data. You would see number of insertions and deletions per person and the plot for number of single nucleotide variation. This is from the project of Undiagnosed Disease Network. I will talk about this. And there's very characteristic relationship between, for example, age of father and number of individual mutations. I mean, we shouldn't be really scared and think about family planning, but generally there is an increase in number of mutations for primarily due to paternal age and also due to maternal age. This analysis doesn't take into account correlation between ages of mothers and fathers, so but you know that both ages are important. So these are primary determinants of the mutational process. So again, the chance that the nucleotide changes in a generation is about 10⁻⁸, pretty small. However, because of the genome is so long, this results in about 70 no mutational events in each class. There are more mutations inherited from fathers than mothers, and there are slightly more mutations in children of older parents than in children of older parents. So that's about mutational process happening in humans. We have variation and we have variation. Okay, so I mentioned to you that I have 2 copies of each chromosome, and I have a variant, right, about every 1,000 bases. How old this variant? Usually, most of this will be very old. When I'm saying very old, it depends, but it can be a few 100,000 years. For example, variant can be 200,000 years ago, some may be older, maybe 400,000 years ago. So you should realize that this mutation happened in possibly non-anatomical human. Right? A lot of variation is shared with for example, with ancient historical hominid species like Neanderthals and Denisovans, right? So these are very old events. These mutations happen very long time ago. And for example, this project GEM is an amazing website. You can look at individual sites of the genome and find out how old they are. What's the genome of the story in the sequence. We have many DNA sequences. We can do inference now, and we can find out about the ages of mutation events and individual combinations of mutations. Old. Why is it so old? Now, think about it, right? So we believe today these are differences. I told you mutations happen all the time. Why do variants persist hundreds of thousands of years? Meaning maybe 10,000 generations, so and so forth. So the process is mathematically analogous to the process of diffusion. There are very small changes in frequency every generation, right? So mutations start as very rare, and they increase in frequency. Some of them, minor fraction of them, increases in frequency from generation to generation by random sampling, and this is very analogous to movement of the scalar particles in the world. That's a very slow process. And this is why they persist. Now, what's important here is that they generate long, but they don't persist very long. So this means that if I'm telling you it's 200,000 years, 400,000 years, pretty much never can be again, right? So you have pretty strong cutoff, even randomly, even without selection events, nothing happening, you cannot be very long. So this leads to the following situation that if I would look at humans from different continents, and I would find a common variant in one group, it's usually also present in the other group. There are very few, almost no examples of one allele representing one continental group and the other allele representing the other continental group. So this exists, but primarily these are specific stories related to resistance to malaria, for example, or some tree. Very few. At the same time, if I would take a few humans from whatever population, I would find common variants, where I would find a position which is constant. I would look at chimpanzees, and this variation will be completely unrelated. Right, so our genetic variation is completely separate from genetic variation of apes, but is absolutely shared among continental populations. Not to say that there are differences between populations, right? But you can, looking at genetic data, you can tell from which continental population this person comes from. But these are due to small changes in frequency in multiple sites, rather than specific allelic markers of populations. Right? So fundamentally, our difference from, for example, great apes is drastically dissimilar to our differences among different populations. So common variants are mostly old, and common variation is shared broadly across human population, human continual populations. Why am I reading this? I already told you this, right? So, okay. What about rare variation? So there is also variation that is very rare, because now we have access to hundreds of thousands of individual genes. And if I'm looking at variation which is found in, for example, in 0.1% or 0.01% of humans, first, there are many variants like this. There are a lot of variants like this. Why is that? Well, first, because these variants don't need this random chance to become common. They are eliminated very quickly from, most of them will be eliminated from the population over time. So they are very young. Second reason they are very young is because of the growth of the human population. We started as a relatively small population back in Africa, and recently, relatively recently, started over exponential growth. It's a very rapid growth. So what the rapid growth means, it means that every generation there is increased number of mutations coming into the population. More and more mutations are coming into the population, and as a result, this is, by the way, if you have no idea how quickly human population grows, it grows very quickly. As a result, we have plenty of variants that are very rare. They're vastly more numerous than common variants, and they are usually relatively new, right? So they've arisen recently within individual human populations, and on the background of very large populations. Again, because of this, for many of the variants, they didn't arise once. Like I told you about the common variation, so the average variant distinguishing microchromosomes, microsets of chromosomes. But a rare variant usually was brought into the population multiple times in high mutation rate sites. Right, so there is a big difference in this behavior. Okay, so this is the variation. Any question about general statistics of variation? Common, rare, age, everything's clear. So how do this translate into genetic differences between human individuals? Phenotypic differences. I'm really seem deprived of that one. So how does this translate into phenotypic differences between the population? So we think that most of the variants have no effect on human biology. So there are changes in DNA, they don't matter. At least this seems so. Right? We don't know for sure, but it seems that they don't matter that well. Still, surprisingly many do matter. So there is a fraction of variants that do matter and do change human biology in different ways. The major focus from the practical standpoint is on specific type of differences between humans as existence of disease. Right, so that's what we really care about when we're thinking about human differences. Right, so some people develop disease and others don't. And there is a clear genetic component to this. Okay, so I'll start with rare variation. I understand that this distinction between rare and common is somewhat artificial because I'm trying to draw a line on the sand. But this is important because there are two fundamentally different regimes, so I will keep this distinction going forward, even though it's not absolute. So some mutations that arise de novo, right, so new mutations that arise in children separated from parents, and variants that are rare lead to monogenic diseases. So the rare diseases very frequently are caused by a single mutation in a single gene. This may not be the full story. There may be other genetic variants contributing, there may be environment contributing, there may be developmental issues contributing, but there is frequently one genetic effect of major contribution to disease. These are some of the patients in our consortium, so we participate in undiagnosed disease network. And what's unique, this is not the largest study of mediated disease, but what's unique about this study is that we have very specific clinical nodes and actually bring patients to clinical sites of the consortium. And we take all comers. So the only requirement is that this disease should be unknown. Right, so this patients lack diagnosis. And there are very many of people that have diseases completely unknown sites. Surprisingly. So this is briefly the consortium. There are 12 clinical sites where the Carnegie Center, and there is of course sequencing core that sequences DNA. The numbers are about 4,500 genomes at this point, including about 1,500 probes. And the disease categories are all over the place. And the thinking, the current epidemiological thinking, is that we have this monogenic diseases, what we call median diseases. Most of them are restricted to very few patients or very few families. Right? Even though collectively, as a group, they affect 1 in 15 people. This may be generous, you usually read these numbers, you always think whether they are inflated or not, but it's probably about right. It's collectively relatively common phenomenon, even though specific diseases individually are very rare. So, This is an interesting point because Mendelian genetics and history of Mendelian disease historically was study of pedigrees, right? It's a study of familial inheritance, how a specific phenotype segregates in the family. Truth of the matter, most phenotypes never segregate in the family because in order to segregate in the family, people have to survive and raise children, right? And in case of many diseases, this is not what people do, unfortunately. So, when the field moved from studying pedigrees, people started collecting individuals and presenting with the same disease, which is a very hard proposition, right? So you have to go and find people who exactly like, and if they are clearly abnormal, and have a common disorder, and you have to sequence and analyze. We also participate in the program trying to sequence patients and families individually, and then you're into this biology guessing game. What's happening now, and what I'll try to convince you, is that we're on the verge of completely changing the rules of the game, right? So this becomes a data science exercise, statistical data science exercise, rather than this fascinating clinical journey. Most of patients come sporadic, sporadic meaning that parents are unaffected, right? So there is no family history. There is a unique case in the family, and parents are generally okay. If it's a monogenic presentation, two things generally happen. May happen. One is it's a de novo mutation, so parents, neither of the parents and nobody in the family carries the mutation. It happens de novo. And a large fraction of our patients have de novo mutations. Again, there's no chance you can find it. It's a Mendelian disease potentially, but there's no chance you can find a study in pedigrees because nobody has the mutation, right? So you don't know the mutation. The other possibility is that this is a recessive case. So there are two alleles, one inherited from father, one from mother, and they together give you disease phenotype. So generally, it can be de novo. So these are two chromosomes in the mom, two chromosomes in the dad, and then there is one new mutation arising. In this case, the child only carries one of the chromosomes with the mutation, so that's a dominant disease, but it's not a dominant access, so we don't see dominant transmission, we see sporadic. Or it's a recessive situation, and mostly what we see cases of compound heterozygosity. Historically, if you're genetics textbook, right, so what you're thinking that recessive disease means that you inherit the same disease mutation from mom and from the dad, and they don't express the phenotype because you need two copies to be affected, and you inherit the same copy as a patient, and this is why you see disease. So this happens very rarely. That happens very rarely in modern data, in rare disease. Usually, in all cases this happens, parents are remotely related. So every time we detect these cases, parents will be remotely related, either due to cultural norm in this community, where frequently, for example, first cousin, third cousins will marry each other, or great-grandparents or great-parents will be from neighboring villages, and everybody will know that there is remote relatedness, but you look at the DNA, and there's clearly a chunk of DNA that wasn't carried by the same person. Mostly what we see, a overwhelming majority of cases we see, are compound heterozygotes. So what we call compound heterozygotes, it means that you inherit one mutation from your mom, one mutation from your dad. They are different mutations, and they arose separately, independently, but they take the same gene. And this is how compound heterozygotes works. Okay. Now the major claim of changing the world, right? So we discussed that Mendelian disease comes from pedigrees and families, it's individual patients and small groups. Now it's all about data science. We live in the 21st century, we have plenty of data. And the genome is a finite place. It's in one genome, right? It's 20,000 genes, it's a lot of genes, but it's still a finite place. It means that if we're going to collect patients with something very rare and unusual, not even something specific, not even restricted to specific systems, but we collect enough of them, we will start finding individuals with mutations within the same gene again and again. Now what I have to do, I have to develop statistical and computational methods that would be based on two ideas. One, they would prioritize places where mutations important for disease would happen. And B, they would tell me, oh, in this many individuals, seeing 3 mutations in the same gene is a statistically significant observation, I can trust it. And of course, what you can do, you can look at these individuals and see whether they have the same disease or different disease. And that's what we're still doing, working with clinicians rather than artificial intelligence. What is interesting about the cohort I briefly mentioned, we have only 1,500 programs, but they all have unique extreme presentation. We found about a dozen genes with recurrent de novo mutations. Nobody knew, right? Because people look at them individually one by one. And the only way to find out, you have to look at everybody collectively. And as soon as you know that this is a statistically significant observation, and this individual shared de novo mutation in the same gene, and they look alike, and they have the same disorder, potentially this gene causes the disorder, and this is how we discover now new genes and new diagnosis by simply looking across data sets. In order for this to work, somebody has to collect sufficient number of individuals with Mendelian disease, rare monogenic diseases, unknown, undiagnosed, and analyze them collectively. So I think we should join forces. And I don't know whether there is a program in Turkey, but I would say it should be, right? Because this is the way to find all of human disease genes. Okay, one observation, and this is nothing in biology is completely true, right? So it's very hard to make statements like this. Of course, there may be 5%, 10% exceptions. We don't really know how many. But so far, most of the mutations we find in monogenic cases impact proteins. Either individuals lack the gene, or individuals have changed in gene function, or changed in the protein that either make it function more, function less, malfunction, or genes improperly assembled in the process we call splicing, right? So because genes are constructed from pieces of DNA sequence going into RNA sequence, and this is where splicing happens, and then we produce proteins. How do we know? How can we build these computational methods and predict which mutation is actually important for disease? About 20 years ago, we started, alongside some others, this idea of doing this computation on it, looking at evolutionary data, sequences from different organisms, right? Because we would think that this protein should function in different organisms, so changes that are permissible everywhere, and changes that are not permissible are not permissible everywhere. And we can look at different functional annotations and protein structures and make some computational prediction about the important population. Now, of course, it's a very big playground for machine learning developments and for collecting more data. This is some form of author of this paper that it just came out of Illumina. It's called QuantLab. The method called Prime AI 3D. This is one of the examples. There are many new methods coming up, and we'll see how well they are going to be adopted. But it has a couple of advantages. One is the sequence of primate species, with an obvious idea that everything what segregates in primate species is probably benign to human proteins. Right? It doesn't happen. And then we're talking about artificial intelligence and language models and various versions of ChatGPT in the space, which is some of that is similar architecture, by the way. And the knowledge of protein structures, which we can now again predict by artificial intelligence, by machine learning, and again looking at evolutionary signals, right? So this is that of the methods that are now being developed and applied, and this probably will grow, right? So we have, again, we have statistical significance of the observation, seeing mutation in multiple patients. And we have a computational technique trying to predict which mutations are important. And I usually talk about computational work because my work is computational, but we should also mention that there is very rapid progress on the experimental side. And in this case, it's presented by deep mutational scanning, and deep mutational scanning is the idea that you physically generate sequences of a lot of variants for one protein, and there is some phenotypic readout, which is molecular readout. So you're trying to test how well the protein works and select the variants in various types of experimental assays. So we now also have techniques. We as a community, we have not my lab doesn't do anything like that. But we as a community, we have tools to introduce very large number of mutations to protein sequence and test them. You cannot do it on the protein scale. Right now, you have to do it individually for each protein, but this is roughly what's done. At least for individual proteins, we have this knowledge. So combination of this together probably would solve mediated disease, at least protein coding proportion to mediated disease. Now we have complex phenotypes. We have diseases that are not rare, that are common, and here I'm much less optimistic about the speed of all progress. But this is a fascinating problem. So we discussed that some phenotypes are mediated, but most of traits you measure and the differences you measure between individuals are quantitative or binary, and they don't segregate in any mediated fashion. They are not extreme outlier phenotypes. They represent some differences in different traits, or maybe slight differences in disease risk, like type 2 diabetes or myocardial infarction. Over 100 years ago, there was this debate between mentalists and some of these individuals participating in the debates, and Fisher who actually resolved it. There was a big discussion whether genetics follow the biometrical laws of these quantitative observations or follow Mendel laws. And biometrical laws include statistical observation, right? Like, for example, normal distribution for height of the population. Or dependency, so the reason we think is genetic, dependency of children's height on parental heights, as we discussed in the beginning. In 1918, the fundamental paper by Fisher actually proposed a model that we're still using today, and this model suggests that there are many loci, right? So many individual variants that are slightly pushing phenotype up or down, and each of those is segregated in mediated fashion, but collectively they result in this observed normal distribution. And for binary traits like disease, we usually statistically also assume that there is an underlying statistical trait, and going over the threshold produces this binary outcome, right? Literally the same one. And early observations suggested that there are very many loci, very many variants involved in complex traits. And this is not a human example. This is an example of experiment on maize, right? Selection experiment on maize going over 100 years. And we see linear constant response to increase in concentration of oil, right? So people have been selecting maize for high oil concentration per seed, and this concentration increased where the low line decreased. And this picture is obviously inconsistent with 1 or 2 or 3 genes underlying the oil content, so it seems that there are substantially many genes. For people in the audience, I expect a completely different crowd for this, so I know that you've already seen a lot of the slides. Maybe I wouldn't do another stand-up here. Okay, so now what's happening? We can actually look at the genes and find out how many loci represent the variation, or responsible for the variation. There are advantages of technology that enable us to learn about DNA variants. We learn about them primarily in the form of genome-wide association studies, trying to relate variation to phenotypic differences, to find a loci that statistically are associated with phenotype. So those that differentiate short individuals and tall individuals, individuals with diseases like diabetes and myocardial infarction or disease-free individuals. And what we now know, we know that there are more than 12,000 loci associated with human height, and there are still loci to discover, so there are probably tens of thousands of loci involved in the height. Still means most of the genome is not involved, but it's quite a lot, it's much more than people used to believe. And common diseases like schizophrenia, type 2 diabetes, Parkinson, Alzheimer's, they also are determined by very large number of genes, and they are called highly polygenic. This is the inference of the effect sizes and median effect size of the variant and number of loci. So what's happening is most of risk for common disease is driven by variants that are common in the population. Because they are common in the population, and there are very many of them, by necessity means that the effect sizes of individual variants are very small. I mean, you can measure them. And what you see here is that the effect sizes of risk are about 1 or 10,000 or 1,000 of standard deviation, right? And for height, this translated to fraction of a millimeter. And number of loci with effect greater than the median are in thousands or tens of thousands for various phenotypes, and the total number of loci are probably higher. So we're talking tens of thousands of genetic signals underlying this. Very weak effect. So, majority of polygenic variation is due to common variants, not rare variants. And many of them are small effects, so this there is a new version of this plot in the literature. This is minor RNA frequency from 0 to 50%, right? So this is what's the frequency of the allele, which is smallest of the two frequencies in the population. And what you see here is that most variants identified by genome-wide association studies that are common are very small effect. And the only variants of large effects are exceedingly rare. Now, majority of mutations, again, this is not 99%, but this is probably 95% of associations identified as involved with common phenotypes are not located in protein coding genes. That's a stark contrast with Mendelian genetics, right? So in Mendelian genetics, mutations are in proteins, and in polygenic phenotypes, most mutations are outside of genes. What do they do? Tell you the truth, we don't know, right? Philosophically, this is a very interesting situation. Because originally you think about scientific progress as somebody studies basic science and then goes and applies it, right? So people study properties of cell cycle, they try to figure out how cell cycle works, and then they go and apply it to cancer research. They're saying, oh, cancer is very relevant to cancer. Or people, I don't know, finding electromagnetic waves, and then build a radio, and then build a cell phone. And that's the idea that there is basic science and then there is applied science. This is a very interesting example because experimental biology would have never discovered this origins, right? Because you cannot produce a lab experiment measuring such tiny small effect in this vast quantities. So what happened is we studied disease genetics, and studying disease genetics, collecting enough samples, there was this realization that we have thousands of very small, minuscule variants, and this is what determines risk of disease and differences in human genetics. Right, so this was the only way to figure it out. So now we have to move back and move to experimental biology and actually find out what they do. But there are some observations that are very helpful. What are those observations? We think, for reasons that I'm going to mention in a few minutes, and also because this is the only logical possibility, that this variance, S is missing, variants, they regulate quantity of protein products rather than their function, right? So they don't change your protein, they don't eliminate your protein, but they regulate how much protein is being produced. So they are very variant. So this is the model. So if you have a gene, right, so gene is okay, everybody's same. Then you have a variance, so some people are different from others, and if this variant is encoded in regions and produces large effect, some variants, some individuals are different from others. Now, if you have a variant in a regulatory sequence, the protein is the same, but the amount of the protein is differs, and individuals are quite similar to each other, but still a little bit different. Now, in our recently published work, there are some issues with this model, but that's the basic model I think the community operates on. 1, 2, 3 is technology working, or it's not working. Working, perfect. Okay. Now, what can we do? What we can do, and what the community does, we can look where this variance reside. It's a complex proposition because there are a lot of correlations along the genome. We have to develop statistical models to figure out what regions of the genome inform risk to disease. Many of these don't bring you very close to biology, but are highly informative. So, for example, in the recent issue of Science, there is this result of Zoonomia consortium, and we can look at the bases that are conserved, are identical between humans and many of the mammals. Turns out this is enormously informative about locations of where variants affecting human disease are. So evolution teaches you a lot. The problem is, we know that these variants are common to all the mammals, right, conserved between humans and mammals, but we don't know what they do. So the analysis of conservation points you to the right side of the variants, but it doesn't tell you what biological mechanism explains this conservation. You can do different things. You can say, okay, if I look at non-coding DNA in humans, usually DNA is chromatinized, right? So it's bound with other proteins. And these proteins generally maintain structure of human chromosome, chromatin. But there are certain areas where some regulatory important proteins that bring signals about the amount of gene product you're going to produce, they have to bind somewhere. And where they have to bind, DNA should be open, it shouldn't be closed. So for example, we had collaborated with John Stamatopoulos's lab in Seattle and some others. So John is, I think, primarily pioneering this technique. So you can look at regions of open DNA, right? Open from chromosome. And the way you do it, you cut it with some enzyme and see what enzyme can cut and test it naked DNA versus chromatinized DNA. Now this suggests that these regions are regulatory. They are important for some protein binding and potentially regulating other genes. Again, we don't know the mechanism, we just know that DNA is open. And we know that there are a lot of signals a lot of GWAS signals here. John and Mark Moran led the paper, I authored the paper with the first observation like that in 2012, over 10 years ago. There are also modifications of chromatin, right? So not only keeping chromatin open, but also changes in chromatin, and they point to variety of specific regulatory roles. And again, we see that many of the variants causing disease happen to be in the regions of modified chromatin. So we can identify that. For example, there are a bunch of projects. So this comes from Digital Roadmap, where we were happy to participate in this consortium that scanned in a lot of cell types. It scanned various chromatin modifications, and you can now see in which cells chromatin disease signatures are enriched. Many papers doing this, so I will have a picture from Shomer and Chattery and Gosha Trinka paper. That's an old paper, but I really like the figure. Many other papers don't have figures that are so clean. So what is clean about this figure? So you're looking at enrichment of genetic signals associated with disease versus annotation, open chromatin, in different tissues and cells. And what you see, if you look at cholesterol, you see that the enrichment exists in the liver. And you never told your method that liver is the important tissue for cholesterol. So we know that this is the true signal, right? It finds liver by itself. And if I look at rheumatoid arthritis, I'm finding immune system as I should. And if I look, for example, psychiatric, neuropsychiatric disorders, I'm finding relevant brain regions. And for example, in type 2 diabetes, I'm finding both pancreatic islet and liver as being important as it should be. Right? So it's not that I'm finding this variance associated with human disease, a very, very small effect. But I'm finding them distributed throughout a big fraction of the genome, but very unevenly. And they are specifically found in regions of regulatory chromatin, and this is not the artifact, because I'm eating always the right tissue type, the right cell type. Right, so this is where we are. And I think that's it, because I don't know what else do we do. We have a lot of consortia trying to experimentally manipulate with the regions. We can look at expression data. For those of you who will stay for more scientific versions of these lectures, tomorrow we'll talk about that. But this is what we're trying to do. We're trying to figure out what we do. Well, thank you very much. Thank you. Yeah. You know what, again, I'm a little bit confused about the format. So I think this is the public lecture format that's supposed to be more general. Yes. The brief question is, we're looking at single cell data now. Single cell attack is an amazing method. And the question is, what would justify moving to single cells, right? If you believe that bulk tissue data are not informative. Where I currently stand, I think bulk tissue data are quite informative regarding position of regulatory regions. I don't think they are informative about expression signals. So I believe that combination of single cell attack and single cell transcriptomics might be a way to figure out if there is context specificity, fine grained context specificity of expression. And I think like using single cell attack data is one of the annotation tracks we should be using. And many groups do like it. If you read some of this, I can include some of our early single cell work in tomorrow's lecture.
Any other questions?
Yeah. Sorry?
What is the recurrence? You mentioned
Oh, the recurrent mutation, right. So recurrent, what I call recurrent it means that you observe multiple events. Right? So in the context of finding gene responsible for mediated disease, I would find de novo event. De novo means that I have a child who developed a mutation differentiating this child from parents in a gene. And then I would have another event in my data set, this different position maybe, but same gene, and another event in the same gene. If I have enough events, and if I have the correct statistical model, I can say, oh, this is not a chance. Right, so this means that these people, like I have more people in my population with severe mediated disease that have mutations in this gene, probably this gene is involved in something. And then I look at these people and see if they are similar to each other or not. And if they are, then I believe I discovered a new syndrome, right? And this is what we do now in the computer rather than. Oh, no. Yep.
Hi, thank you for this informative lecture. It was really useful.
It's redundant to the others, so I if it was informative, but if it was, thank you. Yeah.
Sorry. Hi. Is it working? No, I can yell if you want. Okay, I'm more like into the plants, so I'm more into the plant genetic mapping. But for animal studies, it was a really nice presentation for us. I just wanted to ask you, in the class studies, it's more like a selective type of study, like you are trying to find the best model, I guess, as far as I studied. But in human studies, it's more like a risk statistics, I think, as far as I understand from your lecture. I just wanted to ask you if will it be possible, I know ethically it's a little bit confusing, but will it be possible that a selection might be happening in the embryology section with using GWAS or TBAE?
Okay, that's a loaded question. So a couple things. One is in terms of plants. A lot of the activity that happens in plants is very similar to what I described, right? So people have wide associated studies and organosis. A lot of the same methods are used in the plant world.
Yes.
Now, in terms of the application, and this is specific to agriculture, right? So that's probably less specific to Arabidopsis basic genetics. In agriculture, you use genetics for selection. And I was talking about polygenic prediction this morning. This is what the main game is. So in humans there are two applications, right? So one is if you can predict genetic component. In medicine you predict disease rather than you select on disease. That's answer number one. Answer number two, do we have companies who suggest to do embryonic selection of humans based on polygenic risk scores? The answer is yes. Do I like the idea? The answer is no. For multiple reasons, and the reasons are pleiotropy, that you don't know the effect on other traits, there is not very much of statistical power in this, there is a lot of noise, and I frankly have no idea why to do it, right? If in the mediated rare disease world, that's a different question, and we'll see where the community will go. But I don't want to be cornered with questions about medical ethics, because I'm not an expert. But there is some activity in the selection question, which a lot of people dislike it. But the prediction of risk and there is also a very soft version of selection in rare disease case, and that's family planning. Right, so that's definitely happening as well. And by the way, therapeutics, right? So one issue, if you're asking about applications of human genetics, finding targets for therapeutics is the major practical application, right? Because if you find the right biology, you find the right pathway, you find the gene, you know it's working in the human population, not in the experimental system, not somewhere else. We have genetically informed therapeutics target, and that's super important. Yeah.
You talked about prioritizing the genes and variants. And you said that you can use statistical tools and AI models for prioritizing. I would like to learn about in the rare disease conditions, those tools may change. Are they changing in rare disease conditions?
Changing meaning what? Changing in time, like developing, or changing? So this is my. I'll try to. Again, I'll be talking tomorrow about that more. But in simple version of this, right? So I think what's happening. I think there is a shift. The idea is not brand new, but it's coming to fruition. I think we have some way to go. The idea that you can use statistical methods in rare disease by finding multiple corpus of mutations. So what you have to do, you have to come up with a lot of data sets. Usually statistical methods have two components. One is you have to assign statistical significance that what you see is not random. Right? Because for example, if I have 50 people and I see 3 of them have mutations on the same gene, that's a very unusual observation. But if I collected 10,000, then it's not an unusual observation anymore, right? So I need some statistics to control. The other one is I cannot look at every mutation in the genome, there are just too many. So I need a way to bias my search, right? So change the search. And at least what we do and what other groups do, you take this idea where mutations that are likely to change protein function and therefore likely to cause rare disease, where are they located? Which genes are more likely to harbor rare disease, and that's usually evolutionary thinking, right? So genes are the most selection pressure, and numbers I see in my data set. That's okay. So this combination is just going forward. There is a separate development that I didn't mention that's very orthogonal. Is to use AI models on the clinical side, phenotypic individuals, right? So saying that what is the presentation and what happens with people and try to do the inference, basically try to make the machine do clinical inference. And that's very different, that's not what I was talking about. And that's also being developed quite aggressively. Did I answer your question or I didn't understand it?
So it seems that there are no more questions. We can take break up to tomorrow, but first let us please thank